Well, I think we'll get started. Hello, everyone. Welcome, and thank you for attending today's webinar on the development of in vitro intestinal model with macrovascular endothelium, the comparison of animal-based and xeno-free ECM. My name is Christopher Oak with the Well Bioscience, and I will be your moderator, along with our guest speaker, Scarlett Zodinger from the University of Graz. During the webinar, you can ask questions using the Q&A tab, and we will collect them and present them during the Q&A session at the end of the presentation. Also, we have polls during the webinar, so feel free to participate. Now, I would like to introduce and welcome our speaker, Scarlett Zodinger from the University of Graz. She currently works in Professor Eva Robleg's lab at the Institute of Pharmaceutical Technology and Biopharmacy, where she's working on in vitro cell models to study the fate of drug delivery systems. In particular, she aims to develop advanced xeno-free cell-based systems that mimic the intricate physiology of the small intestine. By incorporating complex physiological features, these xeno-free models provide invaluable insights into drug delivery behavior, leading to more efficient and ethical drug development practices. I will now turn it over to Scarlett.
Thank you, Christopher, for the nice introduction. Hello, everybody, and thank you for attending my webinar today. I'm very happy and excited to be here, and would firstly like to thank the Well Bioscience with John Huang and Christopher Oak for the opportunity for me to present my work here today for you. I'm a PhD student at the University of Graz, as Christopher mentioned. I'm working and focusing on the development of in vitro barrier systems, and today I will be talking about the development of an in vitro intestinal model, considering the macrovascular endothelium and the comparison of animal-based and xeno-free ECM systems. I want to start with the physiology of the intestines. The intestine is the largest and most multifaceted organ in the human body and functions as the main site for drug and nutrient absorption. The small intestine consists of 4 layers and can be divided into 3 parts, namely the duodenum, the jejunum, and the ileum. The innermost layer, the mucosa, is characterized by evaginations and so-called plicae and villi, which increase the surface area for drug and nutrient absorption, and is also characterized by tubular invaginations into so-called crypts, which provide a protected site for stem cells. The mucosa is lined by a single columnar epithelium consisting of various cell types, for example, the epithelial enterocytes, mucus-producing cells, and stem cells. A closer look reveals that the intestinal epithelium is underlying an extracellular matrix. This matrix consists of 2 layers. The first layer is the basement membrane, representing a thin layer directly underlying the intestinal epithelium. This consists of 2 polymer networks, namely the collagen 4 and the laminin network, which are interconnected through proteoglycans. In addition to the basement membrane, the second layer, there's also the interstitial matrix. This interstitial matrix hosts blood vessels for drug and nutrient absorption, as well as immune cells, fibroblasts, and various structural proteins such as the collagens, the elastins, and proteoglycans. Through integrins, these are mechanoreceptors, the extracellular matrix is in constant contact with the epithelium, enabling inter- and intracellular signaling, as well as biochemical and chemical variations. Due to the complexity of the small intestines, various systems have been designed to mimic these physiological occurrences. The most best known models are so-called 2D models. These are usually consisting of 1 or 2 cell lines cultivated on plastic or glass surfaces. The most used and best known cell lines representing the small intestinal epithelium are the CARCO2 cells and the goblet cells HT29 MTX cells. Both cell lines are derived from human carcinoma and show high similarity with the small intestinal physiology of humans. Both cell lines need to be cultured for up to 3 weeks to reach a full differentiation and full functional model. In addition to 2D models, there are also so-called 3D models. These models can be divided into organoids and spheroids and into models that use scaffolds or hydrogels as a structural basis underneath the cells. For models which use hydrogel as a structural basis, often the Matrigel, for example, an animal-derived substance is used. As a third approach, there are dynamic systems using bioreactors, pumps, and microfluidic systems to mimic flow conditions or motility to simulate, for example, peristaltic movements. With this in mind, our aim of the study was to develop an in vitro intestinal model considering the villus epithelium with the epithelial and the mucus-producing goblet cells, the ECM and the endothelial cells to also mimic the systemic absorption. Further, it was important to develop an animal-free system to exclude any unwanted interactions, as animal-derived hydrogels are often not precisely identified with regards to their ingredients. Batch-to-batch variations can occur, leading to unwanted interactions when brought into contact with human cells, for example, leading to immunological reactions. This brings me to my workflow. In the first step, we evaluated animal-free gels for the simulation of the ECM, which have similar rheological properties as the human ECM. In the second step, we tested the cell growth on these ECM gels and investigated the effect of the gels on the cells. In the third step, we used an endothelial cell line EAHU926 to culture them on the basolateral side of an insert via inverse cultivation. Combining the above-mentioned steps, the model was established considering the villus epithelium with the epithelial and mucus-producing cells, the ECM, and the endothelial cells. And finally, the model was characterized with regards to their mucus formation, morphological structures, for example, the formation of tight junctions, as well as cell differentiation. Furthermore, permeability studies were performed with defined drug groups. ECM substitutes that are most commonly used are collagen 1 and Matrigel. Both are derived from animal sources, which may lead to unwanted interaction, as mentioned earlier. Collagen 1, for example, is derived from rat tails, while Matrigel is one of the best known and mostly used ECM hydrogels is an extract from mouse tumor cells. Both have to be processed on ice due to premature chelation when handling at room temperature. In addition to these animal-based systems, there are also synthetic hydrogels, namely VitroGel or Peptigel. VitroGel, for example, is a polysaccharide-based hydrogel, and Peptigel is a peptide hydrogel. Both gels can be processed at room temperature and do not need any extra cooling, and are modular, which means they can be combined with other proteins or substances and can be diluted to achieve a desired composition or viscosity. Thus, we decided to test Matrigel, VitroGel, and Peptigel as an ECM hydrogel for our development. In the first step, we characterized all 3 gels with regards to their rheological properties. The studies were performed with a dynamic viscosimeter. All gels were studied with regards to their dynamic viscosity, the thixotropy, and the viscoelastic ratio. This was followed by studying their effect on the cell growth of the co-culture CACO2 and HT29-MTX cells. Therefore, these cells were cultivated in a cell ratio of 7:3 in transvals, and on the gels, as previously shown by our group. After 7 days of cultivation, cell proliferation was studied using an MTS assay. As the intestinal tissue is under constant pressure due to peristaltic movements, the ECM substitutes for in vitro development needs to withstand certain shear forces. If you look at the dynamic viscosity, all 3 gels show a starting viscosity in the same area between 79 and 103 Pascal seconds. And they show shear thinning behavior above a shear rate of 7.5, which is in accordance with data found in the literature. In addition to the dynamic viscosity, we also investigated the thixotropy. The thixotropy is a measure for the recovery of the inner gel structure after complete destruction. Here, we found that VitroGel is the only one to recover by about 80% after complete destruction. Matrigel and Peptigel only reached a recovery of 30 and 40% after complete destruction, leading to a partly destroyed inner structure of the gel. These results indicate that VitroGel would withstand the intestinal stress caused by motility and in the event of destruction would recover to about 80%. The cell proliferation studies of the co-culture consisting of CACO2 and HD29 MTX cells were performed by an MTS assay. Here the results showed that the cell proliferation on Matrigel and Peptigel were comparable to the cell control or slightly higher. In contrast, proliferation on VitroGel was increased by a factor of 1.7. This means that a confluent cell layer is built more quickly due to the increased cell proliferation, thereby accelerating the cell differentiation. An overall comparison of all tested parameters, including the rheological characterization and the effect on the cell proliferation, as well as taking into account the handling of the gels, led to the conclusion that we move forward with the VitroGel hydrogel system. The VitroGel hydrogel system has the big advantage of quick and easy handling, showing in vivo relevant rheological properties, and also enhances cell proliferation by a factor of 1.7. Another advantage is that it can be processed at room temperature and does not need additional cooling. And furthermore, it is based on polysaccharides and is not animal-based, leading to reproducible and consistent results. I'm sorry. In the third step, following the characterization and identification of an ECM substitute, endothelial cells were cultivated on the basolateral side of an insert. Therefore, the endothelial cell line EAHU 926 was used. For this, we used an insert of a transvaal system, put the insert upside down in another plate, which enables the addition of endothelial cells on the basolateral side. After 2 hours of incubation, the insert was placed back in the original plate, which allowed adding the VitroGel hydrogel system and the co-culture of CACO2 and the goblet cells. This system was cultivated for 7 days, followed by the characterization via tear measurements and visualization using histochemical investigations. The results show that the endothelial cell cultivation on the basolateral side of the filter was successful. The tear value after 1 week was 15.7 ohm square centimeters, which is in accordance with the data found in the literature and indicate the formation of a confluent cell layer. These results were further verified by immunohistochemical investigations, which can be seen in the right figure. Here, it is shown that the endothelial cells formed a confluent cell layer after 7 days. In blue, the cell nuclei, and in green, the cytoskeleton. Considering the above-mentioned steps and combining them, the in vitro model was established with considering the following layers. In the first step, we cultivated endothelial cells on the basolateral side via inverse cultivation. This enabled the addition of the VitroGel hydrogel system on the apical side, followed by seeding the co-culture of CARCO2 and HD29 MTX cells on the ECM hydrogel. This system was cultivated in the transfer system for up to 2 weeks and fully characterized in the final steps. The in vitro model was characterized using various methods. First, morphological investigations were performed. For this, the mucus layer, mainly produced by HD29 MTX cells, was visualized using ecridine orange to identify a confluent mucus layer on top of the epithelium. As cell signaling is enabled via cell-cell contacts, so-called tight junctions were visualized using immunohistochemical staining. CARCO2 cells or the epithelium form so-called tight junctions, which enable cell signaling and also form a barrier. For the visualization, occludin, which is a protein of the tight junctions, was stained and visualized and imaged via confocal laser scanning microscopy. And lastly, a confluent endothelial cell layer was identified via fluorescence microscopy. In addition to the morphological investigations, the cell differentiation of CARCO2 cells in this model was tested. Therefore, the release of the brush border enzyme alkaline phosphatase was determined. When this enzyme is brought into contact with nitrophenyl phosphate, hydrolysis occurs, which can be detected by 405 nanometers by absorbance measurements. And lastly, permeability studies were performed with regards to the paracellular transport route, the transcellular transport route, and lastly, the activity of the PGP-F-mediated efflux transporter was investigated. The characterization of the model showed promising results. In the first figure, you can see that a confluent cell layer was formed on top of the epithelium. The confluent or the mucus, you can see here stained in red, whereas the cells are stained in green underneath. The mucus mainly produced by HD29 MTX cells is comparable to the human small intestine. As cell signaling is enabled by the cell contacts, the tight junctions, more precisely occludin, was stained. The figure illustrates a confluent cell layer. In blue, you can see again the cell nuclei, in green the cytoskeleton, and in red the connected cells via tight junctions. Further, we can see the typical microville structure of CARCO2 as indicated by the white circles. The last figure depicts the endothelial cell layer on the basolateral side of an insert. The endothelial cells formed a confluent cell layer and proper tight junctions, as again indicated by the red staining. Apart from the morphological investigations, the differentiation of the CARCO2 cells was determined. Cell differentiation was determined through measuring the release of the enzyme alkaline phosphatase, short ALP. It was found that the model reached 95% ALP release compared to a CARCO2 monoculture, and this was achieved only after 14 days of cultivation. Compared to that, a co-culture of the CARCO2 and HD29 MTX cells without gel only yielded a value of 61% compared to the monoculture. The slight decrease from the model is due to the lower number of CACO2 cells in the model due to the co-culture of the epithelial and the goblet cells. As a last step, permeability studies were performed in the model. Therefore, different transport routes were tested, namely the paracellular transport route between through the intercellular space using atenolol, that's transcellular transport route directly through the cells using antipurine, and lastly, the activity of the PTP-mediated efflux transporter using rhodamin 123. The results are depicted are shown in the right table. Here, the apparent permeability coefficients, shortly PAP values, can be found for all three tested substances. Atenolol reached a PAP value of 0.6, which is in accordance with data found in the literature using excised small intestinal tissue. Antipurine PAP values were consistently high in the model, with its peak after 14 days and the value of 33.6 for PAP value, also comparable to values found in the literature for in vivo small intestinal tissue. And lastly, the PTP-mediated efflux transport activity was determined via rhodamin 123, as can be seen here. The PTP-mediated efflux transporter led to the transport of rhodamin 123 back into the luminal side, and we were not able to detect an apical to basilateral permeability. So in summary, all three tested drugs were found to be comparable to ex vivo and in vivo data. This brings me already to my conclusions. In summary, the results show that VitroGel is a suitable substitute to be used as an intestinal ECM substitute. One of the major advantages is that it is an animal-free product and exhibits a high recovery and in vivo physiological relevant rheological properties. Further, it increases the cell proliferation of the intestinal cells by a factor of 1.7. The cultivation of endothelial cells was also successful. This facilitated the establishment of the in vitro cell model, including the epithelial and the mucus-producing cells, as well as the ECM with the VitroGel hydrogel system and the endothelium to mimic systemic absorption. The characterization of the model confirmed the confluency and accelerated intact cell-cell connection, cell differentiation, and the formation of a mucus layer, while enabling ex vivo comparable drug permeation, all of which could be achieved within 2 weeks compared to the standard cultivation of 3 weeks. And a quick teaser on what we'll be doing next. Next, we'll be incorporating other key ECM components, such as collagen or laminins, with using the VitroGel high concentration hydrogels. Thanks to the mix and match system of the VitroGel hydrogel systems, the ECM can be designed to the desired composition and rheological properties. Additionally, we want to add immune cells to get closer to the anatomical physiology. And lastly, we were going to test the Vitro prime cell culture inserts within our model. This brings me to my last slide. I would like to thank my supervisor, Professor Dr. Eva Roblek, and the members of our research group, Drug Delivery and Advanced Manufacturing at the University of Graz, and my friends for their support and guidance. Thank you very much for your attention, and I would like to hand over again to Christopher.
Thank you. That's a great webinar for you, Scarlett. Again, we have an extra slide here for contact information for the Well Bioscience. You need more information about our hydrogels, what our products do, please visit our website. And also you can scan our LinkedIn We do have product announcements, updates about product productions and new webinars coming up as well. I'm going to run one more polls, and we're going to actually go to Q&A session. If you have any questions for Scarlett about her work, please put it into the chat right now, and we'll go into the Q&A session. Okay. Okay. One of the questions here is, how does the VitroGel polymerize? And that is a great question. The VitroGel hydrogels do not polymerize based on temperature change. It is based on the gelation of the ionic cross-linking. So when you mix with the cell culture medium, the salt molecules from the cell culture medium will help the hydrogel cross-link. There is no additional cross-link reagent needed. So the whole operation is at room temperature. So it makes it more for a more consistent operations and for consistent results on your ECM workflow. Okay. How were the permeability studies with drugs performed, Seringa?
Yeah, so we established the model, as I mentioned earlier, considering the co-culture, the ECM, and the endothelial cells. We waited until full differentiation of the cells, which we tested prior to the permeability studies, and then added the 3 different tracks in the apical side, so on the luminal and epithelial cell side, to test the permeability through epithelial cells ECM down to endothelial cell side to mimic the systemic absorption, and also tested the direction against the systemic absorption to test, for example, the PGP activity. We evaluated the data then via HPLC measurements to calculate the permeability coefficients, if this answers your question.
All right. A couple more questions here, and let's start to come in. Another question from Patrina. What kind of media did you use? Did you add any component to the media to promote differentiation?
Actually, no, it's a good question. We used the standard Dulbecker's modified Eagles medium, the common medium for cultivation of the CARCO2 and HT29 MTEC cells and endothelial cells. And no, we did not add any special substances. It was just a commonly used DMEM medium for cell cultivation.
Great. Another question is, she wants to thank France wants to thank you for a really interesting work. He would like to know, why did you choose a cell ratio of 7:3, CACO2 to HT29?
This was actually based on work from the same working group right now at Professor Roblek's lab. Within this publication from 2013, they tested various ratios of these 2 cell lines to get a mucus layer similar to the physiological occurrences, as well as tear measurements. So it was Schimpel et al. From Molecular Pharmaceutics. So yeah, I also cited it. It can be found in the internet. But yes, it was previous studies from the working group.
Thank you. Suzanne has the questions. How did you explain the site of application of ethanol leads to different permeabilities? If it's exclusively paracellular transport, the direct should not matter and similar values should be received?
Yes, it was quite surprising to us too, because ethanol is supposed to be, according to the literature, paracellular transport drug. However, we found that the ethanol also showed activity for the PGP-mediated efflux transporter. So that's the reason why we did not get similar values for the apical basilateral way compared to the basilateral apical way. So that's the reason. So it's not exclusively paracellular transport, but also a little PGP-mediated efflux.
Another question from Peach. In the physiological situation, the blood goes through the EC and mucus cells to the top intestinal chamber, and then back through the mucus cells and EC layer to the bloodstream. In your model, did you confirm the media really circulate like that?
No, we did not. But that's. I'm sorry, I have to read the question again. Through the mucus cell. No, we did not perform these measurements, but that's a point we have to look into.
Okay.
No, I'm sorry.
Not a problem. All right. I don't see any more questions here. Anybody else have any questions, please send that. If not, we'll give 2 more minutes and we will end the webinar. Someone has a question here. Trina. All right. Trina, what is that question? Let me see here. Okay. Another one is I am working with Ali cultures using human Norman colon organoid-derived cells. We were able to increase goblet cells and enterocytes following differentiation. I am interested in incorporating the endothelial cells. Would you be willing to share your protocol for doing that?
Yes, the protocol or the publication based on this webinar can be found. I cited it and can be found under my name. So it's a recent publication from September 2023 and will provide an SOP protocol later on.
Okay, great. Any other questions? We don't see any more. So again, thank you everyone, and thank you Scarlett for that presentation and great work on your research here and your team there. And thank you Eva Roblek for allowing us to have you join us and presenting. Everyone have a great day. And then again, we will send you a link to the webinar recording as following this as well. And we'll send you more information about how to find all the information, the publication and protocols and applicable products that was used. Have a great day, everyone. Thank you.
Thank you.